Fetal brain development begins early in embryonic life, commencing around the third week of gestation. The process initiates with the formation of the neural tube, a critical precursor to the central nervous system. By the 8th week, the rudimentary brain structure takes shape, dividing into primary vesicles that later differentiate into distinct regions. Over subsequent weeks, neural cells proliferate, migrate, and differentiate, establishing the foundation for the intricate network of the developing brain. This ongoing maturation involves key milestones such as synaptogenesis and myelination, ensuring the progressive formation of a fully functional fetal brain. You want to know how and when does the fetus starts developing the brain? Continue watching, but before, share, comment on this video. Also, subscribe to our YouTube channel and don't forget to click on that bell icon to get regular updates on our new videos. The development of the human brain begins early in embryonic life, specifically during the third week of gestation. The process is complex and involves several stages, ultimately leading to the formation of the fully developed brain. Here is an overview of the key stages in embryonic brain development. Neural tube formation, weeks 3-4. During the third week of gestation, the neural plate forms from the ectoderm, which is one of the 3 primary germ layers in the developing embryo. The neural plate then folds to create the neural tube, a structure that will give rise to the central nervous system. Neurulation, weeks 3-4. Neurulation is the process by which the neural tube is formed. It involves the folding and closure of the neural tube. The anterior, front end of the neural tube develops into the brain, while the posterior, back part becomes the spinal cord. Formation of brain vesicles, weeks 4-5. The anterior part of the neural tube expands and differentiates into 3 primary brain vesicles: the prosencephalon, forebrain, mesencephalon, midbrain, and rhombencephalon, hindbrain. Development of secondary brain vesicles, weeks 5-8. The primary brain vesicles further divide into secondary vesicles, giving rise to 5 distinct regions: telencephalon and diencephalon. From the prosencephalon, mesencephalon remains unchanged, and metencephalon and myelencephalon from the rhombencephalon. Formation of neural crest cells, weeks 4-8. Neural crest cells, a group of cells that migrate from the neural tube, play a crucial role in the development of the peripheral nervous system and various other structures. Cell proliferation, migration, and differentiation, weeks 8 onward. Neural progenitor cells within the developing brain undergo rapid proliferation, migration to their designated regions, and differentiation into specific cell types, including neurons and glial cells. Synaptogenesis and myelination, throughout gestation and postnatally. Synaptogenesis, the formation of synapses, connections between neurons, begins in the fetal brain and continues after birth. Myelination, the process of insulating nerve fibers with a fatty substance called myelin, also occurs throughout gestation and into early childhood. The intricate process of brain development is highly orchestrated and involves a combination of genetic, molecular, and environmental factors. Any disruptions during this critical period can potentially lead to developmental abnormalities or neurological disorders. The timeline provided here is a general guide, and individual variations may occur. Share, comment on this video. Also, subscribe to our YouTube channel and don't forget to click on that bell icon to get regular updates on our new videos.